So, Mohsen, are you okay if the meeting is recording?
That's fine by me. I
Okay.
just need the sharing permission. I cannot share my screen. Yeah.
So, well, it's an honor for me to introduce Mohsen Sadeghi. So we have the honor of sharing same office and same work group for some time. He's really one of the world experts in biophysical membranes, in the theory and simulation of membranes in biological context, and I'm really looking forward to what he has to show us today. And yeah, sure, please go on, Mohsen.
Thanks. And yeah, that was too nice an introduction. So let me try to share my screen and hopefully you can see my slides now.
Yes.
Okay. So yeah, there is quite a bit of them. So I will give you a weighted presentation on some of the topics I will probably spend more time. The rest we can sometimes skip. So in general, I'm going to talk about mostly a membrane model that I've been working on for the past a few years and what we could do with it and how it helps us study large scale dynamics of biomembranes. Okay, so for those of you who are not familiar with membranes and bilayer membranes, this is a very brief introduction, but probably it's known to most of all of you that these are basically bilayer self-assembled structures formed by lipids. And the reason that this happens is that the lipids have a head group, which is hydrophilic, and tail, which is hydrophobic, and they prefer to keep the hydrophobic parts together. So among other forms of structures that can achieve that is a bilayer. And this is something which has found functions in living cells, offers a mechanical and chemical barrier to the cell, and also on top of that can help with the regulation of input and output, participates in signal transduction, like in neurons, and so it's a vital part of the living machinery. But from the point of view of biophysics, biomembranes are really interesting because they can have a variety of geometries and topologies, and there is the most important part of the biophysics is that they are rather rigid structures. So at their scale of action, they are basically more or less behaving as solid sheets for out-of-plane motion, but on the other hand, the lipids inside the bilayer, they have the freedom to flow freely, so with a certain viscosity. So they are 2D fluids, but in 3D, they are basically solid sheets with a rather high rigidity. And it's in principle difficult to shape membranes into very curved geometries. So that's why usually when inside a cell you see something with a high curvature, like the endoplasmic reticulum, you would look for a reason that would keep the membrane in this basically high curvature region. And that reason is usually proteins, which actively help shape and preserve the shape of the membrane. I will talk a bit about how we can also study this as well. And in general, at least for the past 40 years, people have looked into different ways of modeling membranes, spanning a wide range of scales, both in space and time. And so you can start from doing all atom simulations with the different compositions of the membrane, basically model the lipids and their original resolution, or you can try to coarse grain this, come up with rougher representations of individual lipids. And as you move along this curve of scales, at some point, basically you lose the resolution to resolve individual lipids, and that would be usually the realm of continuum methods. So the model that I'm going to talk about, it basically lives in the scales which are accessible by continuum methods, but it's a combination of the two. So it's still a particle-based model, but shares a lot of features with continuum-based approaches. And the same hierarchy of scales also applies to the proteins that I talk about, that they basically sculpt the protein, sculpt the membrane. So you can also have fully resolved proteins and study the reason that they would induce a certain curvature in the membrane, or you can just assume that the membrane basically reacts to a certain simple topology of the proteins and preserves a certain curvature. Or you can altogether get rid of this representation and only work with curvature fields, for example. As you probably can assume, that the approach that we would take to coarse graining depends on a lot of details, as what's the resolution that we are interested in, and what's the performance that we want to gain in the end. Because, for example, if you do an all-atom model of the membrane, you're very much limited to very small patches, usually sub-micron size, and in terms of the trajectory lengths, you can't gain something as much as microseconds, and that would be that would be an unlucky day. So we can start to do the bottom up as I described. So start to coarse grain from a all atom representation of the lipids. For example, this middle one would be the famous Martini force field, which was originally developed for lipid bilayers. And there are a zoo of other models of using different number of beads per lipid and studying how this would self-assemble and the membranes that they would build, but they are no longer correspond to a known basically chemical species. But on the other hand, we can basically be done with the whole bottom-up thing and try to look at the membrane from top down, from the continuum surface basically, and try to come up with models that can give us the mechanics that we expect from that. By the way, if it's okay if at any point you interrupt me and ask questions, so if anything is not clear, feel free to ask. And so the model that I'm going to talk about, this is something that I developed and worked on for the past couple of years, is basically a particle-based model formed by these dumbbell-shaped particle dimers, and they are in a closed packed assembly. You can think of it as a so in-plane hexagonal lattice, and they only interact with their nearest neighbors through bonded interactions, and they also have out-of-plane angle bending between these in-plane and out-of-plane bonds. And we try to parameterize this model so that it would reproduce the mechanics of the membrane to the resolution that is possible. And to give you a sense of scale, in this figure down right, I have basically produced one protein. This is a Shiga Toxin protein. I will talk about it later in the talk. And so it's basically up to scale on top of the beads that form my model. So you can imagine that's a rather highly coarse-grained approach.
Mohsen, when you do this model, this is, I guess maybe you're gonna say it now. Is this the top-bottom approach or the bottom-up?
This is the top-down approach, yes. So this would be my parameterization strategy. So On the continuum front, so if you consider the membrane as a continuous surface, you can work with the curvature elasticity. And with such a particle-based model, we would have a discrete geometry of distances and angles, and these two could be connected through differential geometry of the surfaces. Also, for a continuum-based model, you have elastic constants, which is encapsulated in this very famous free energy model, the free energy of health, the Herflich functional basically of the membrane energy. So this function would give you the energy density at each point on the surface based on the mean curvature, which is the H, and the Gaussian curvature, which is the G. This is a model from the 70s, but it has been working perfectly in modeling the mechanics of the membrane. So this is now the standard way of approaching this. And people can measure this kappa and kappa bar constants experimentally for different membrane compositions. And so we know everything that is to know about the mechanics just through these two elastic constants, at least at the scale that this functional is applicable. And from a particle-based perspective, we have a usual force field. In this case, Morse interactions for bond stretching between one particle and its neighbors, and a harmonic angle bending. And these could be added up, and an effective energy density could be calculated on this hexagonal lattice of particles. So ideally, these two would match. So there would be an energy equivalence between a continuum energy density and this particle-based force field. On top of that, I also consider the possibility that at some point a particle is occupied by a protein, an agent that induces a certain curvature in the membrane, and would also potentially change the local elasticity. So,
Can I ask a quick question?
sure.
So when you present the force field, you say actually you need this Morse potential.
Mm-hmm.
But why can't you just do a harmonic bond or even a fixed bond? So I mean, is there a reason for that?
No, you're absolutely right. We have the freedom to choose other functions for the force field. This is something that I've been comfortable with using. It gives good results.
Okay.
Also, it is basically a breakable potential. So we can also model membrane rupture with this.
Okay.
But that's it. If you're not interested in that large deformation, you can work with harmonics.
Okay, thanks.
And so, yeah, when we have the protein on top of the membrane, at least virtually, we would have the Helfrich functional for the pure membrane and introduce an indicator function where proteins exist to add up something else, and an energy density for the proteins. And this is basically what I have done using a rather simple optimization technique. So these two plots, the surfaces are comparisons of the energy that you would get. For example, in the top plot, this is for a particle of pure membrane. We have the Helfrich with the blue wireframe and the effective energy density that comes from the force field in the red wireframe, and this is the best match. And the surface is in the basically in the space of curvature. So locally, I am looking at the energy that would exist for different combinations of principal curvature. So these are curvatures in two principal directions, and in an optimal case, the free energy density should match for all the range of curvatures that we are interested in. And in the plot to the left and bottom, so this is the case where I have also included a protein with a certain elasticity on top of this bead. And so you can see that the energy landscape is a bit shifted, but we can also again fit another set of parameters to that. So we would have families of parameters for different lattice parameters of the model for different regions, and we can also mask the force field to switch on and off the presence of the protein.
Uh, One first question was, so if let's say there's no protein, just the matching between the particle-based model and the continuum, you have these different parameters that you have to match, this k, this kappa and kappa bar, and then the d and the other parameters. So to match them, is this you do a numerical thing, to a numerical simulation to match them, or some kind of numerical calculation, or is an analytical result?
So it is numerical, but I'm not doing a simulation per se. This is done before, basically. So this is done in the space of, so it's a parameter space optimization. What I do is that I have a representative volume element, basically a small enough model of the membrane for which I can dictate a certain curvature. And so that differential geometry basically tells me that if I dictate these two principal curvatures, what would be the resulting distances and angles, and what would be the resulting energy density that comes from the force field. And then I can basically iteratively optimize for that. But this is not done on a large simulation. So hopefully when I do it for this small element, this would also propagate to a larger simulation. I hope it makes sense. So if you remember, I also mentioned that the bilayers are basically fluid in plane. So we need to also do something for the fact that if we keep on with the fixed topology, if the particles are only connected to their neighbors, they cannot go anywhere. This would be a complete solid. And so to introduce a certain fluidity in plane, what we do is basically we allow for these bonds to be flipped. So one diagonal could be flipped to the other, and one particle can basically through time change its neighbors. And in order not to disturb the equilibrium distribution in the model, we need to do that through a Monte Carlo move. So propose the bond flipping move with a certain frequency, and then accept or reject it based on the change in the energy through Metropolis Hastings scheme. And this is how it looks like, just in plane. We don't have any out of plane motion yet, just to show that indeed using this bond flipping move allowed the membrane to flow. So this is a simulation for a membrane under a gravity-like force. And we can also play with this proposal frequency to change the effective viscosity of the membrane, but I will refer to this a bit later in the talk. So this is a full simulation in which you see the in-plane and out-of-plane motion, and the simulation is when I have put basically a spherical nanoparticle on top of the membrane, and this nanoparticle interacts with the surface of the membrane through the surface adhesion potential. And so what was interesting about this system is that it shows the interplay between the adhesion strength and the bending stiffness of the membrane. Am I still on? So can you still see and hear me?
Yeah.
Okay. So this is a study that we did on this system, and if you change the strength of adhesion, so the bending rigidity is held constant, and we can work with different nanoparticles that interact with different parameter u with the surface of the membrane, and by increasing the u, we can encourage or discourage the nanoparticle being wrapped by the membrane. So there existed a theoretical model in the continuum limit, which would be the red line in this plot, and our simulations gave results which are the dots here. So it's a prediction of this behavior, and in these two movies, you see the comparison between two limiting cases with this basically dimensionless parameter u, which shows the ratio between the energy, the surface adhesion energy, and the bending energy of the membrane. One other thing that we also studied here was the fact that this neck region that is formed should obey a minimum surface description, so it should have zero bending energy, and that would be a catenoid shape. And so the green lines, if they're visible in the plot, are fits of the catenoid. So the model seems to work at this scale of interest, at least in terms of mechanics of bending, even at highly curved regimes. And also, I have basically looked at the case where I have a protein on top of the membrane, and what would be the curvature resulting from different elasticities prescribed for this protein. And yeah, I've also compared with certain experimental or simulation values for these curvatures to show that we can cover the range of interesting curvature inducing agents. But one other topics that I basically looked at after building this force field was the fact that if you fit these parameters to equilibrium thermodynamics of the membrane, or basically the mechanics of the membrane is an equilibrium property, what would happen to the kinetics? So if you simulate this model with an integrator, stochastic or deterministic, how fast would it fluctuate, basically? And this is a general problem, even with all atom force fields, you don't necessarily get the correct kinetics. And this is more so of a problem with coarse grain models, because when you do coarse graining, you're artificially smoothing your energy landscape, and you're usually going towards faster kinetics. And this is very important when you want to look at an equilibrium processes. So if you average out in an equilibrium simulation, faster kinetics doesn't matter, even helps with your sampling. But if you're looking at an equilibrium process, then the kinetics could be a deciding factor. And we know that if you want to look at cellular processes, they would be anything but equilibrated. Yeah, so an equilibrated cell is a dead cell, and this is a dead cell. So how to include the kinetics that you would get for a membrane floating in a solvent to this rather coarse grain model? So I mentioned a bit about the in-plane kinetics. We have these Monte Carlo bond flipping moves, and we can control the frequency to change a certain viscosity, for example. This gives us a level of control over the in-plane kinetics. But as this schematic hopefully shows, there are lots of effects going on when you have two particles or two inclusions in the membrane. They have a certain in-plane mobility, of course, but this in-plane mobility is also affected by the shear contributions from the solvent, and they can also exist crosstalk between these motions. So there would be solvent-mediated and membrane-mediated interactions of the hydrodynamic nature between two particles. So it could get really complex real fast. If you focus on the out-of-plane kinetics, you would definitely need a good description of solvent hydrodynamics. For example, compare it with the simple case of a spherical particle floating in a fluid domain, and we have the famous Stokes-Einstein description, gives you a diffusion constant based on the viscosity of the fluid and the geometry of the particle. My original idea was that, okay, is there a comparable expression that we could use for particles on the membrane, and to basically prescribe a certain diffusion constant for the in-plane and out-of-plane of this out-of-plane motion of these particles. And I came up with basically a general scheme for doing that. And how it works is that we first of all, we consider an anisotropic diffusion for each of these particles, and we try to model the response of the solvent through introducing Gaussian-shaped boundary conditions and then solving for Stokes equations to see what would be the effective response, and then calculate an out-of-plane diffusion coefficient, and hopefully put the two in a consistent framework to do a stochastic dynamics. And that consistent framework is the overdamped Langevin dynamics with hydrodynamic interactions. So this is more or less an equation for Brownian dynamics. And so it's a souped-up version. It looks a bit scary because now, first of all, we have the diffusion tensor instead of a diffusion coefficient. And second of all, as you can guess from this sum, the motion of one particle is also affected by the forces existing on the rest of the system. And this is a description of hydrodynamic interactions. If you push one particle, the rest of the particles are also affected via the motion that is introduced in the solvent. And this also translates into a correlation when you want to calculate the noise term. So the random motion of particles is now correlated through these diffusion tensors and an assembled basically global diffusion matrix.
Can I ask a question, Mohsen?
Sure.
So when you talk about hydrodynamic interactions, well, I mean, I think it's well known that the thermodynamics is not affected. So your potentials are actually, or your effective potentials are the same whether or not
Yes.
you do this hydrodynamic interaction or not.
Mhm.
Is this obvious from this equation? I can't see it, namely, because I think it seems like, well, not only your kinetics is changed, but also your equilibrium is changing. So how do you actually see this? Is this clear from this top equation that the average is 0? Is that what?
Yeah, you're absolutely right. The fact is that with different kinetics, you should get the same thermodynamics. And how this is derived from this equation is basically the same fluctuation dissipation argument. And the way that we build the noise term related to the friction or the diffusion of particles also guarantees that even with this scary looking equation, everything settles down to Boltzmann distribution.
Okay.
Yeah, that's what's happening.
All right, thanks for clarifying.
And so I basically proposed a general form for these diffusion tensors in this shape. So each particle, so you can consider the diffusion coefficient prescribed for one particle, and this pairwise diffusion tensor which describes the hydrodynamics. For each particle, we have the in-plane contributions and the out-of-plane, the parallel and the perpendicular components here. And for the pairwise interactions, I'm only considering the interaction between the perpendicular terms. So the out-of-plane motion is mediated through the solvent, but the in-plane motion is more or less local. And so this is a certain assumption at this point. We can make this more sophisticated, but that seemed enough for the dynamics that I was interested in. And for the basically now all we need to do is to find expressions for these components. Luckily for the in-plane contributions, we have a very good model of Safman and Delbruck. And so this is something that they worked on in the 70s to study how a cylinder floating in a 2D fluid sheet would diffuse around. And so there is a viscosity of the membrane and the viscosity of the solvent in there. So it also takes care of the shearing interactions between the particles in the membrane and the solvent. And it's a very well behaving model. So it gives the in-plane diffusion coefficient rather robust. For the out-of-plane motion, things are not that easy, and the details of the model, of course, involved. And so I tried to basically calculate the perpendicular components. The way that I did that was to, as I mentioned, introduce a Gaussian-shaped boundary condition. So one of the particles is pushed upwards, if you want to imagine it, with a certain distribution of velocity, or with a certain distribution of stress. So we can do it both ways. We can prescribe a velocity locally and look at the stress that is developed in the fluid, or we can prescribe a stress and look at the velocity field and the basically displacement that comes out of that force. And we can use this information to calculate the friction coefficients or the diffusion coefficients. And I solved this, basically I solved the Stokes equations for inertial hydrodynamics, which is the case at our scales of interest. And you see here two plots for the two cases where I prescribe velocity and read the stress, or prescribe the stress and read the velocity. And these are different solutions. So this is for a single planar membrane. These are solutions that I found when we have a parallel set of membranes with different separations, as well as spherical vesicles. So I have omitted ugly looking expressions and semi-analytical ways of finding these, but basically the end result is that we know the stress distribution or the velocity distribution for a certain prescribed boundary condition. And the way to use that is when you put a force on the jth particle, you can read the displacement that would be resulting from that force on another particle, and based on that you can calculate a diffusion, an out-of-plane diffusion coefficient. So the force comes from integrating the stress, as you may, and the velocity is basically from, and displacement from the velocity field. And it gives us a certain distribution of this pairwise diffusion coefficient. So you have the self-diffusion values at 0 here, and as you move away from one particle, its influence dies out, usually with an 1/r dependence. But as you can see, the geometry has a significant influence on both of these cases.
Mohsen, one first question. I mean, are you actually solving the Stokes equations together with the membrane? This part was not completely clear to me.
That's a very good question. So again, I want to basically calculate these coefficients beforehand and then use them in the Brownian dynamic simulation. So I want to keep them constant values only depending on the pairwise distance between particles. So in that sense, they are not coupled to local deformations and motions of the membrane. But in deriving these expressions, I used several facts about basically the assembly of the particles that I call the membrane as the boundary of the solution. For example, I've assumed that they are in-plane incompressible, and I have introduced that as a condition at the interface between the fluid and the membrane. Or for example, these Gaussians have a length scale embedded in them. So that length scale corresponds to a size for these particles from the point of view of the solvent, because otherwise everything is in the continuum limits. The solvent wouldn't care if you're moving a small part of the membrane or a large part of it. And yeah, these are the sort of information that I put in the solution. But when that solution is obtained, then we just take diffusion coefficients and run the simulation. And during the simulations, I don't do a recalculation, if that was your meaning. I hope I answered. Okay, then.
I was also wondering about several things. So the first of all, this Rij is the distance between two membrane particles, I presume.
Yes,
But
and
this is
in plane distance. Yeah.
yeah. So it is the effective hydrodynamic interactions over a larger distance.
Yes.
Okay, and then you see all these curves. So clearly, you actually distinguish a single plane from multiple planes or multiple membranes. Is that what you're saying? So
that's true. So depending on the geometry, of course, the solution to Stokes equations are different. So the blue basically points come from a single planar membrane, one surface which is in contact with an infinite solvent domain. And the green and orange one comes from, as you mentioned, basically a parallel set of membranes, yes, with different distances, and that would definitely affect the mobility because now we have a confined fluid in between. And I refer to results which basically correspond to that case. And yeah, so for a spherical vesicle as well.
I mean, what people in the literature often use is, of course, well, Stokes-Einstein, but also the Oseen tensor.
Yes.
They seem to be okay for the flat interface or for a single interface. They are completely off for multiple interfaces. Is this what you or multiple?
That's a very keen observation. Yes, that's true. I have shown in the paper that we wrote on this that so for the case of single planar membranes, we can obtain an analytical result with this method, and you can show that for particles separated long enough, then it basically settles to an Oseen tensor description. So locally it deviates a bit, but for the far field, it's basically the same thing. Yes. But of course, when the geometry changes, then the Oseen tensor is no longer applicable.
Okay, very interesting. Thanks.
So, firstly, this is an easy investigation of in-plane motion, basically. So here we don't see the hydrodynamic of the solvent, just the result of that Staff-Mandelbrot model in combination with the bond flipping moves, which are still there. And what I want to show is that we can successfully calibrate this. So this is at this point just a calibration for the in-plane diffusion, and we can get diffusion coefficients for these quote-unquote proteins on top of the membrane, which are comparable with the experimental values. Also, the viscosity that the surface viscosity that comes out of the model also is in the range that people have measured in experiments. So we can calibrate the model for good in-plane behavior. But what we are really interested in is the out-of-plane kinetics. And I wanted to look at the dispersion relations for the membrane fluctuations in the out-of-plane direction. So I use a reference model to compare the result to. This is the model developed by Udo Seifert. And this is a very comprehensive model, at least in the continuum-based regime. So it uses the Helfrich energy to model the bending of the membrane, uses the Oseen kernel for the hydrodynamic interactions, but again, as a continuous kernel, so it integrates over the surface of the membrane without any mention of the length scale. Includes the fluctuating lipid density, so the free energy that it uses also includes a term for two leaflets and the internal compressions of the leaflets. And it uses the area difference elasticity between the two leaflets. Also, there is a friction between them, so if basically they have different expansions or they want to move with respect to each other, they experience a certain dissipation. And it basically boils down to a linear model for which when we obtain the eigenvalues of this time evolution operator, we can get the time scales and the frequencies basically. the motion prescribed by this. So I did simulations of membrane patches in thermal equilibrium and mapped the height function. So this is what people call a Mohsen description. So you just assume that the membrane is a surface, and the height of the membrane is a well-defined function of x and y in this case. Then we can get a Fourier transform of this and look at the different fluctuation modes in Fourier space. So the first line is the equilibrium result. So we know that the distribution of power between different modes obeys this 1/Q4 relation. This is a very well-known result of the Helfrich function. And so in the plot to the left, you see this equilibrium thermodynamics description, and the points come from the simulation. So no surprises there. They follow the black line, which is what is described by this first equation. The interesting thing about this plot is that now we can see that the membrane actually reproduces the mechanics that we put in parameterization. So this is the same kappa coefficient used for the elasticity description. So this now comes full circle. We use this in the parameterization of the force field, and now we have used this force field in the simulation, and now we can basically get the kappa as the output here, which is nice. So the mechanics are reliable. But more interestingly is the plot to the right, which now looks at this dispersion relation. So the kinetics that is prescribed by the model plus the hydrodynamics. So this bimodal decay or bimodal relaxation comes from the Seifert model from the previous slide. And so these are the dashed and dot dashed lines in the plot. I have checked the case for two different interleaflet friction. So this is the only unknown parameter in the model. The rest of them are basically either membrane properties or the solvent properties that I have manually put in the parameterization. But the friction, this is not something that I have modeled in the model, in the membrane model. So I've checked for two cases of values for this parameter B, and you can see that for one of them we get very good fits for the both modes. So the top mode, which doesn't care about the friction, is the actual hydrodynamic mode. This is the dissipation in the solvent. The slower mode or the slipping mode is the internal dissipation of the membrane, which of course is sensitive to hydrodynamics. But the interesting thing is that we also get a certain dissipation from this model, which comes from the internal density fluctuations, which corresponds to a certain value of internal friction, basically. So as I basically claimed or mentioned, we need the kinetics, especially for non-equilibrium cases, and it is only fair to also put that to test. And one interesting non-equilibrium steady state case which comes into play when we have membranes is where we have non-thermal forces acting on it. So one interesting case that people have looked at extensively in experiments and then also in the theoretical models is when this protein, the bacteriorhodopsin, exists in the membrane. So it's an integral membrane protein, and it's light sensitive, so it absorbs light, and what it does is that it pumps one proton across the membrane, so helps basically get this ion through. And so bacteria use this protein for a rudimentary form of photosynthesis. So they basically absorb the light energy through this gradient of protons. And what happens is that when it lets one ion through, it exerts a small force on the membrane. That's what makes it interesting from a membrane biophysics point of view. And if I'm remembering correct, people call this the purple membrane. So yeah, you can make this also in a lab. You can introduce this bacteriorhodopsin to a membrane and then shine a light, and you can observe the effect of these forces. So I built basically a two-sided pump model for this protein. So we have particles representative of the existence of a bacteriorhodopsin, and they can push or pull the membrane based on the state of the pump. So the state of the pump is more or less a theoretical description. So these pumps have a direction, a certain direction. They always pump the proton in one direction. But if we have a certain concentration of pumps which are oriented in two opposing directions, we can get an up or down force on the membrane. And so there is a very well-known stochastic model for that. We are looking at this type of three-state reactions that the pump can go from the off state to the down or to the up with a certain prescribed rate. And then we can look at the kinetics that comes from the action of this, basically. And so I have looked at the mean square displacement that the existence of these pumps would induce in the membrane. So in the blue curve, we have the equilibrium case without these active forces. The orange curve is when the pumps are basically active. And you can see that it very visibly shifts the distribution of the squared height on the membrane. The theoretical model gives us so this first one comes from just summing up the 1 over Q4 expression. And no surprises here. The values, the numerical values matches what we expect more or less accurately. The second relation is basically in the active case. So this corresponds to this the three-state model. And this is an interesting fact. So this the capital lambda here is the Austin tensor. It is the Austin tensor, and so it depends on the viscosity of the solvent, and this is an averaged mean square displacement. So a property which is basically in itself doesn't seem a dynamic property, depends on the dynamic a kinetic property of the system. And this is something that you observe in non-equilibrium cases. That's why it's important to have a good description of the solvent behavior. Also, one hallmark of this non-equilibrium processes is that the distributions now become non-Gaussian. So the non-Gaussianity is something that people look at to see the effect of the action of these pumps. So I've compared these two properties basically between the simulation and what is expected, and we get surprisingly good match for the mean square displacement prescribed by this model and what we get from the simulation. And we could also successfully show that there exists an observable non-Gaussianity due to the action of these proton pumps. And finally, to apply this to basically a real life example, a more sophisticated example, I considered a red blood cell for which we have a combination of at least two of the geometries that I looked at. So the leaflets that are facing outside are considered as free-floating planar membranes, but the inner leaflets are basically experiencing some crosstalk with the other side of the cell. So this obeys the expressions for parallel membranes. And so I put in a variety of properties. I don't go into detail here, but this is a field which is also has much recent interest in. So people are interested in doing single cell diagnosis. They put this one red blood cell under a phase shift microscope, and they can study how it fluctuates and they can get 3D models of its shape. And this is what I did. So I asked one of these groups which did these experiments to give me a mesh representation of the form of the red blood cell. I used that mesh representation to build the model, and then I compared the result with the fluctuation spectrum that they got from the phase shift microscopy. And these are the results. So I'm not sure if that's clear enough. So in this movie, you see a fluctuating red blood cell.
Mohsen, one first question. The fluctuations are the height fluctuations, you mean, that with respect to, like, let's say, the normal shape of the red blood?
Exactly. Yeah. So what we look at is the thickness fluctuation. So add up the basically the height change between the two side of the cell. So we measure a mean thickness over the cell. So these two gradient contour plots are basically the mean thickness distribution from the simulated case and the experimental case, and then we look at how this thickness changes through time. And so with this coarse-grained model and basically the efficient way of tackling the interactions and the hydrodynamics, I could do a very long simulations. So I looked at the fluctuations for 150 milliseconds. Still, it's barely reaches the experimental range, but it's enough to observe the behavior. So this is the power spectral density for these thickness fluctuations. And I've compared the results from the simulation, the blue dots, with the results from the phase shift microscopy, the orange squares. So they show a very good match of behavior across the frequencies, and this is obviously a kinetic result because we are looking at how fast each of the modes are basically fluctuating, and it's a function of how you're putting the hydrodynamic model for the different components of the system.
Why is it that your simulation cannot reach lower frequencies?
This is basically a limitation of the trajectory length. So, yeah. The fact is that yes, if we use the whole trajectory, maybe we could get some results up here, but then that would be very unreliable. So I chopped up the trajectory into parts to get better statistics, and that's why I'm a bit limited in terms of frequencies. And then when we have a functioning model, we can also play with different properties of the cell. For example, the cytosolic viscosity or the extracellular viscosity, or the membrane viscosity of the red blood cell. In this case, I'm showing that the cytosolic viscosity has an observable effect on the kinetics, and this is something that also has been of interest in this experiment. So, for example, when someone has diabetes, that affects the hemoglobin concentration inside the cell, and that would be observable in the fluctuations of even one red blood cell. And so we also showed that the same effect could be observed in this model. Okay, now let's move to a case where we are using this. So up to this point, I've just introduced that we can also parameterize for the presence of proteins, but I didn't really show any results for that case. So this is an interesting example that I looked at. This is a protein called Shiga Toxin. This is the same protein for which I showed that scale comparison figure. This is the same protein that was residing on the surface of the membrane. So Shiga Toxin, the way it works is a two-part protein. So it has a subunit A and a subunit B. The subunit A is the actual toxin. If it gets inside the cell, it disrupts protein production. The subunit B is basically tasked with getting the subunit A inside. And it's a very interesting protein because people have observed that if you put this on a GUV, on a bubble of membrane, it can, in time, form this very long tubules inside the vesicle. So it has a passive entry mechanism. It doesn't need any machinery actively getting it inside. When it's on top of the membrane, it basically just uses the membrane mechanics to get inside. The working theory for how it works currently is that these proteins get together through some interactions, some membrane-mediated interactions, so to say. They can form pits and eventually long tubules just based on the curvature that they locally induce in the membrane. People have also looked at this in empty simulation. What's the effective curvature that comes from the interaction between the subunit B and the membrane? And this is something that I can use to compare just in the parameterization phase. So I already showed you this comparison, unfortunately, when I didn't talk about the Shiga Toxin. So I told you about the fact that I can parameterize the protein also for producing a certain curvature. So this is the result of a simulation for the presence of these proteins on top of the membrane. And so here I've omitted the membrane particles. You only see where where one protein is supposed to be, or where the force field is basically masked to produce the presence of the protein. If you look at the way that these proteins move around, you can also see that they tend to form some aggregates, at least temporarily. They prefer to stay together. So it hints at a certain interaction between these particles. While I haven't put in any direct interaction between the particles that are basically representing proteins, they only interact through what has been prescribed by the force field. So these are purely membrane-mediated effects. And it would be interesting if we can separate this and we can measure the potential that would exist between these particles. So in general, there are two types of membrane-mediated forces that could exist, and there is a huge amount of literature on this. If you're interested, we could have forces of energetic nature. So this comes from the fact that the membrane prefers to minimize the free energy that, for example, is modeled by the Helfrich function. So in the case of these rotationally symmetric proteins, the membrane prefers them to be as far apart as possible. There is no good solution or no minimum shape solution when they are in finite distance. So it translates to a repulsion. On the other hand, there are forces in action from an entropic origin. So people call these thermal Casimir forces. The way that they work is that if you have inclusions or proteins that could suppress the fluctuations of the membrane, they would be pushed together to minimize the entropic expense of this suppression. So if they are dispersed everywhere, they would basically stifle long wavelength fluctuations. But if they are grouped together in an island, the membrane has more configurational entropy. This is a rather lengthy derivation, but the resulting force to the leading power has this 1/R4 dependence. So it's an attractive force. And usually you see an interplay between these two natures when you're looking at membrane-mediated forces. So I'm not going to explain this in very minute details, but I'm using basically a 2D fluid mixture model. I'm looking at the in-plane fluid, which is formed by the membrane particles or these protein particles, and they can. So we have interactions of the membrane nature, or the membrane protein nature, or the protein nature. So it's basically a fluid of two types of particles. And there are good descriptions of this in the dense fluid literature. And so I used a direct correlation function described by this Ornstein-Zern accumulation and the hypernetted chain relation closure, so to say. So this expression shows that if you prescribe the potential, what would be the resulting pair correlation function. And that's all there is to it. So it's kind of an inversion. And so I did this for basically on the trajectories that I obtained from these simulations, like the movies that I showed you. And I could gain an effective interaction between proteins only. So I have taken out the membrane-membrane interactions from this results. And you can see an interesting, basically, potential. So it has a considerable amount when they are close together. I've compared it with the attractive force model, with the entropic model, and of stifling inclusions, and also with the repulsive force coming from the energetic nature. It has some similarities, but it seems that I'm predicting a bit deeper potentials that is predicted by the attractive model. Also, there is a certain dependence on the stiffness of the protein, and this is something that I also studied through the kinetics of the clustering mechanism. So there is this classical way of looking at particles that come together and form a cluster. And people have come up with distributions of cluster size based on the rate of formation and dispersion. And so these are not very important details. I've just used my own interpretation of the number of ways that the cluster of these proteins can be formed on top of the membrane, and found out a certain probability distribution that we expect to exist for the cluster sizes. From the trajectories, I use an off-the-shelf algorithm to identify these clusters based on the pairwise distance between particles. And here you see the maximum cluster size as a time series for these different protein stiffnesses. Again, you see a strong dependence. So as we expect for the proteins that had higher interaction strengths, we would get larger clusters in time, and smaller for the other cases. And so on the bottom, you see a fitting of the probability distribution that we derived based on this kinetic aggregation theory, fitted to the distribution of cluster sizes obtained from the experiment, obtained from the simulation. There exists a very good fit, so that's a good model for this case. What we can use from the model is this parameter which acts like a chemical potential of the formation of a cluster. We can also see that it discriminates obviously between the stiffness of the protein. And also what was really interesting for me was the fact that we can also compare. So the model includes the possibility of one macroscopic cluster to form, so the so-called sol-gel phase transition. So in the long run, you can get some of the particles basically being removed from the rest and joining just one island. And this G star parameter is the fraction of particles that would contribute to this large cluster. And this also matches the maximum cluster size that we observed in the simulation very well. So this is a very successful observation of basically the matching between this clustering theory and what we obtained from the simulations, for which we have not included any direct interaction between the proteins. And finally, I wanted to look at also the free energy landscape of this clustering dynamics. I defined a continuous reaction coordinate based on the inverse of pairwise distances between the particles. And I'm using the weighted histogram analysis method, the VAM method, to look at the effect that the surface concentration of these proteins have on the probability of them getting together or being dispersed. The reason that I can use this free energy method is the fact that I am not adding new particles when I change the surface concentration, but I'm just masking the force field. So in the end, everything can be interpreted as a bias in the potential. And this is basically, I've looked at the fact that this bias also correlates with the surface concentration of proteins that I expected. And these are the actual free energy landscapes. So I can look at them across this reaction coordinate. So as we move towards the right, the higher values of Q, this would correspond to more aggregated states, the other way around dispersed, and the vertical axis is basically the surface concentration. And we can see a varying behavior based on also the stiffness of the protein. So for this very successful case, for which we had the largest clusters, we can see that the minimum of the free energy persists more or less as we increase the surface density. So whatever was aggregated remains aggregated for higher concentrations. But as we increase the stiffness of the protein, there is a significant shift. So for stiffer proteins, when you move to higher surface concentrations, they prefer not to form large clusters. And at least my argument for the reason behind this is that for stiffer proteins, we also have higher repulsions between the proteins coming from an energetic origin. So there would be an interplay between those two energetic and entropic effects that I talked about. We can also try to see what's the optimal stiffness for the protein if we are just interested in getting a highly aggregated state, and it basically gives us a certain value for which I can compare the resulting curvature to the experiments. I don't have experimental results for the actual value of the protein, but yeah, it's interesting that at least the curvatures match with what we have observed. And this is also another case for which we don't let the proteins aggregate on their own, but we start from a already formed large cluster. You can see that they induce a rather highly curved pit in the membrane. But what was interesting for me is that they tend to more or less preserve their proximity. So if you compare the displacement of this protein cluster particles forming this protein cluster and the actual particles of the membrane, in comparison, you can see that this cluster really persists a long time. So the reason for that, on top of the interactions that already exist between these proteins, is that when this highly curved region forms, this also has a stabilizing effect. So if the proteins want to escape this pit, they need to go through a region of opposing curvature, and this is unfavorable, of course. So they tend to remain in the proximity of the pit. So I also wanted to look at or investigate if my claim as to the entropic nature of these attractions also makes any sense. So I calculated the distribution of entropy as a function of that reaction coordinate that I defined. So this is another parameter, but the small s is related to Q. So it also, when it increases, it means more aggregated cases. When it decreases, it means more dispersed ones. And I've looked at different cases with different surface concentrations. And the interesting thing is that for lower concentrations, I can see an observable increase in entropy when particles aggregate. And this is in complete compliance with what we expect from the entropic forces of this thermal Casimir nature. But when we move to higher concentrations, we also need to include the loss of entropy due to limited configurational states possible when you want to aggregate proteins together. So there is an opposing effect, and we see it's working when we move towards higher surface concentrations. Actually, proteins prefer not to be aggregated based just on this entropic effects. Yeah, I also looked at the entropy in the space of membrane configurations to make sure that this claim has some correlation to the fluctuations of the membrane. So I tried to directly measure the entropy based on the probability distributions in the model space of these fluctuations. These are a bit preliminary results, so take this with a grain of salt. The interesting thing that I observed was that we actually have these two islands of higher entropy and higher aggregation, and more or less lower entropy and lower aggregation. If we compress it just across this reaction coordinate, you can see the expected behavior that as we move towards higher aggregations, we get an increase in entropy. And that was it. Thanks for your attention. If there are questions remained, I would be happy to take them. And this is a rather old photo of our group. I'm not sure. Yeah, Maurizio should also be in there somewhere. Yes.
Yes, I am.
Back in there, yeah. and one of rare
Pre-COVID times.
right. Yes, exactly. And one of rare occasions that Berlin had nice weather. Yeah.
Yeah, great. Thanks for the cool talk. Really cool stuff. So anyone has another question or something? I guess past the hour, some people already had to leave probably. So.
Oh yeah, I really stretch on, right?
Well, so I have a question. In the last simulations that you showed, that there was the formation of the clusters bending the membrane.
Mm-hmm.
Like in the simulations, you have to have some boundary conditions because you're just simulating this square, right?
Mm-hmm, mm-hmm.
So what do you do in the boundary so it doesn't collapse completely?
Okay. So we have periodic boundaries in plane directions, in the x and y directions for everything, membrane and protein included. Also, I use a barostat basically for this to sample from a tension-free membrane. So the in-plane components of pressure are basically kept at zero. But the box size fluctuates, but the membrane remains tension-free. I hope that answers your question.
Yeah. Yeah. Okay. So I guess that's pretty much about it.